Hello students. So today we shall study about the albugo, the fungus albugo. Albugo is also called by the other name cystopus. So albugo belongs to the class Phycomycetes. It is the first class of fungi. Subclass Oomycetes. Oomycetes. Order Peronosporales. Order Peronosporales. Family Albiginaceae. Albiginaceae. Example is albugo or cystopus. There are about 25 species of albugo, and all these species they live as obligate parasites on a number of flowering plants which belong to the family Compositae, Cruciferae, Convolvulaceae, Amarantaceae, Caparidaceae, etc. In India, there are about 7 species of albugo, of which the common species is Albugo candida, or it is also called as Cystopus candidus. So this particular species, it causes a disease in the cruciferae plants. Cruciferae plants means the radish, mustard, cauliflower, cabbage, etc. So this albugo candida or Cystopus candidus, it causes disease in the plants like radish, mustard, cauliflower, cabbage, etc., which belongs to the family Cruciferae. This disease is called as white rust disease. White rust disease means, see, on the leaves, so on the leaves of these plants, it causes a shining, white, smooth patches, powdery patches on the leaves. Hence it is called as the white rust disease. So the major symptoms of the white rust disease is white, shining, smooth, mealy patches, which later becomes powdery in appearance. This is the main symptom of the disease. It can infect any part of the plant like leaves, stem, branches, flower, fruits, etc. Except the leaves, it can infect any part of the plant. So what are the symptoms of this disease? See, what happens is the infected part of the leaf, it shows white, mealy appearance, and it shows swelling. The infected portion shows swelling or blisters. Blisters formation is seen and it becomes twisted. This is the main symptom of the disease. The leaves become succulent. Succulent means it becomes thick, and in extreme cases the floral axis, the axis of the flower, it shows the gall formation, and the flower axis, it becomes 12 to 16 times thicker than the normal size. Next, the petals. The petals become, they become like sepals. Sepals means it is the the first whorl of the flower. And the stamens become leaf-like structure. The stamens become leaf-like structure. The entire flower axis, the entire floral axis, it becomes succulent and turns purple in color. So these are the symptoms of the disease. So how to control this disease? We can call it as control measures or the management of the disease. How to control this disease? Firstly, eradication of the infected crop, removal of the crop. And secondly, crop rotation. So the crops may be grown in the same place with two different types of crop so that the disease severity can be reduced. And thirdly, by spraying certain copper fungicides. So this is about the control measures or the management of the disease. So next we shall study the vegetative structure. Vegetative structure or the thallus vegetative structure or the thallus of albugo. So as you all know, the plant body of fungi is made up of mycelium. Mycelium is nothing but a collection of hyphae. So this mycelium is highly branched, aseptate. Aseptate means cross walls are absent, and it is multinucleate, or it is also called as cenocytic. So this mycelium, it enters into the host tissue in between. It is called as intercellular. It grows in between the host cells and it produces globose hostoria, which penetrates inside the host cells and absorb the nutrition. So this is about the vegetative structure of the mycelium. So coming to the reproduction. So reproduction, it reproduces by both asexual and sexual methods. So in this video, we shall discuss only the asexual reproduction. Asexual reproduction in albugo. So it reproduces asexually by the formation of sporangia, or it is also called as conidia. These sporangia or conidia, they are born on a special hyphae which is called as sporangiophore, or it is also called as conidiophore. So let us study this reproduction with the help of a very simple diagram. See, this is the epidermis of the host tissue, epidermis of the host tissue. So just below the epidermis, this mycelium, it grows intercellularly and it produces the globular haustoria. This is the upper epidermis and this is the cuticle. And here what happens is, see here you have the mesophyll cells inside the leaf tissue. You have the palisade parenchyma and spongy parenchyma, the mesophyll tissue. So inside this mesophyll tissue, the mycelia, it spreads intercellularly. Intercellularly means in between the cells, and here and there it produces a globose haustoria to absorb the nutrition. And just below this, this is the epidermis, upper epidermis of the host tissue. And these are the intercellular hyphae. It forms a dense mat of hyphae just below the epidermis. So, when the hyphae matures, it produces elongated, unbranched, club-shaped structures, which you call it as the conidiophores or the sporangiophores. So, just below the upper epidermis, the dense mat of hyphae produces an unbranched, elongated, club-shaped conidiophores or sporangiophores. So, these conidiophores, it cuts off a chain of conidia or sporangia. So, these conidia or sporangia, they are produced in basipetal succession. Basipetal succession means the younger ones at the base and the older ones at the tip. So, these conidiophores, they are separated from, these conidia are separated from one another either by intercalary cells, or it is also called as disjunctar cells. So usually these are spherical in shape, that is globular, thin-walled and multinucleate. But due to the pressure, it changes its shape and it becomes polyhedral or cuboidal in shape. So these are easily, as they are exposed to the atmosphere, during dissemination, during dispersal from the chain, it gets gelatinized and it gets easily disseminated by the wind current. So when these conidia or sporangia, when they fall on a suitable host, it germinates into a new mycelium. So this germination of this conidia or sporangia is of two types depending upon the external atmosphere. When one condition is when there is high humidity and less temperature, then you call it as the sporangia, the sporangial germination. See, this is because the sporangia yet undergoes cleavage into 6 to 8 bits of protoplasm. So each bit, it gets metamorphosized into a kidney-shaped biflagellate zoospore. These zoospore, it swims for some time and then it becomes encysted. Encysted zoospore means it takes rest for some time. It changes its shape. It becomes globular in structure. So later it germinates. It produces a germ tube. This germ tube penetrates the stomata of the host tissue and further it repeats its cycle. This happens when there is high humidity and less temperature. When there is less humidity and high temperature, the vice versa of this condition, the sporangia, it germinates directly, it penetrates into the host tissue directly through the stomata without the formation of zoospores. So direct germination, then the spore is called as conidia.
Indirect germination through the formation of zoospores is called as sporangia. So this is about the asexual reproduction in fungi. So in the next class, in the next session, we shall study about the sexual reproduction in albugo.